Hi, this is B.8 Nucleic Acids. Now, before I do these PowerPoints, I upload a exam question to the website, and I looked over the past four papers to try and find a single question. All I could find was simple regurgitation, and there were only 2 marks, and that was in just 2 of the papers. So either they're too scared to put this stuff in because it overlaps too much with bile, or they're going to hit you with a big structure question, so I would watch it just in case. And not completely skip this topic. So first of all, I want to jump down to one main point here, that these all these things link together, these nucleotides and everything else, through these OH groups here, through condensation reactions, water coming out. So let's use that to help us through everything here. First of all, if you go to the data booklet, which I'll do a bit later, you'll see it'll just be spot the difference. So both of these are labeled and in the data booklet, and if you look carefully you'll see that these are the areas that are different, and so this is missing an oxygen here, so it's deoxy, so it's missing an oxy, so it's deoxyribose, and as I'll mention later, remember to always number it going in a clockwise direction from the oxygen. So this is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. Okay, so on the carbon 2, there's the hydroxyl group is missing for deoxyribonucleic acid. Okay, so here we go into what's more important about the structure. So you look here, 1, 2, 3, 4, 5. So all of the riboses are linked by phosphate groups in the 3,5 position, and you will see that both for these linkages here and for these linkages here, you're losing water, so there'll be OH groups. So if I just jump to the data book, so this is the data book here, and you can see that the linkages here are on. If I write this down, this is 1, 2, 3, 4, 5. You can see that there's an OH there, there's an OH there, and so that's how it combines. And you will also note that these are other options, the OH groups, they're the only other options that you have for linkages to occur. And so there you have the 3,5 bonding for the sugar phosphate backbone. Okay, so just some regurgitation information that's probably not going to be on the test because it's too close to the biology syllabus. So the DNA is double-stranded, it has an alpha helix. Moving on, memorizing ATGC we'll get into in a second. So just a bit more information here. We have an anionic nucleic acid wrapped around basic histones, giving a slightly basic pH of 7.4. And it is hydrophilic, so there are hydrophilic and hydrophobic parts of the DNA, which is also called a polynucleotide, which helps in its stability. So probably remember those highlighted, bolded, underlined bits if you happen to get that regurgitation question on this bit. Alright, now this is a little bit more detail on the structure. Now I remember ATGC based on an acronym that I'm not going to say because it's telling me too much personal information there. It's at a particular place. That's how I remember it. And AU is like for Australia, AU. So I don't know. That's how I remember it. Let's analyze this. So chemically, if you can't remember that, can't think of an acronym, could you come up with that by yourself? And the bit to note is you get a double ring joining to a single ring. I think it's good to escape from here and just go to the data booklet because that's what you're going to have in the test. So going to the data book here, this is what you'll be looking at in the test situation. So you can see that you have these double rings, and you've got to work out which one they join up to. And if you're a very visual person, you might just remember those two at the end are joined up together and then these go to there. That could work for you. But there's also the look at the fact that they're joined by hydrogen bonds, which is joint N, O or F. Join the hydrogen from an N, O or F joins to a lone pair electron. And so if you look here, you've got a situation here where these could form hydrogen bonds, and if there's a lone pair there, and so that looks like it could match perfectly with here. There's a lone pair there, there's a lone pair there, and that could match up. So there's a lone pair there to go to here and here, and there's a hydrogen here to go to here. And so that looks like it matches up. Okay, and for this situation here, there is a lone pair here, there's a hydrogen sticking out here. That's not a hydrogen bonding one. That one there, 1, 2, 3, 4, that could possibly have a hydrogen bonding to it. So there's only singles, they're all broken up, so it's most likely to be this area here. So this looks like this would have the 3 bonding up and this would have the 2 bonding up. So we just need to find a situation over here that has the opposite of that so they can pair up nicely. That's the hydrogen that could link up to here. And there's an oxygen that could link up as well. And ditto over here, off in sort of a different direction. So it's highly likely that it is a double matching up to here, either with this one here or this one here. So you can sort of guess that these two really only fit with the adenine. So you could chemically have a good guess and work it out based on the 3D structure of what could actually stick together to form hydrogen bonds to stick those two DNA chains together. So back to the PowerPoint now. So I've described to you how you can work out that these things are linked up in this situation with hydrogen bonding, how you can just chemically work out its AT or GC. And then obviously the U is sticking in there as well because only the U has those 2 bonds. So it's important to remember it's stuck by hydrogen bonds because that'll help you work it out as well. Alright, so the nucleotide is the monomer, and that we've discussed before. These are the only areas where there's OH groups. Alright, and this one just happens to be sticking out on the end. So these ones in a 3D structure because they stick out at the end, they allow it to project out here, join up to the other DNA strand. Okay, so moving on from that, what's the difference between DNA and RNA? Basically the ribose is a deoxyribose, so there's a missing hydroxyl group, and the base, it's not AT, it's AU, so you could get a question on that. Codon or a triplet code or a codon. So every 3 nucleotides codes for a specific amino acid, and that here is the code, just to let you know that we know what it is. It's actually a legit thing. And so if there's 3 U's, that'll be phenylalanine, I think that is. And so that's one regurgitation question that was for some reason worth 2 marks. Not even, you'd think it'd just be one worth 1 mark, but no. Alright, and so the regurgitation question that I've covered on the past paper, and that's up on the website after these videos, is GMOs. Sorry, GMOs are when the DNA of one species is inserted into another in order to do several things. And I've just regurgitated this. As you can see, most of the crops now for corn or cotton are GMOs. So I've just regurgitated this out of a textbook. Basically, you don't want the crops to die, but you can also increase the amount of flavor or nutrition or resistance, or supply vitamins and vaccines and all sorts of stuff from GMOs, even producing pure fuel from algae and all sorts of stuff going on. A lot of things we don't know, such as, is there a long-term effect? Will it the gene spread to the entire population? Does it cause allergies? Lots of unknowns here. That's probably good to read those because you might not have thought of some yourself. Okay, and so that's it for this topic. I would just have a quick look at some memorization points and hope that they don't give you a huge structure question, because they don't do that very often. Not for DNA. Okay, thank you. Bye-bye.